My name's Catherine Howell. I'm a neurologist at the Royal Children's Hospital and a clinician scientist fellow at the Murdoch Children's Research Institute. My area of expertise is epilepsy, and particularly severe infantile epilepsies. Now, these are really severe seizure disorders. Babies having many seizures every day, and they're often resistant to treatment. They're much more than just seizures though. 1 in 6 babies will die before their second birthday. The majority of survivors will have significant developmental impairments. The longer that seizures are uncontrolled, the bigger the impacts on development. So stopping seizures promptly is of critical importance. But being able to stop seizures relies on understanding the causes of these conditions. Now, 35 years ago when MCRI started, we didn't know the causes of these conditions. We knew about the conditions, but not what caused them. It turns out there's a variety of genetic causes, but we didn't know that at the time. This is Levi. He started having seizures when he was just 1 day old. He had many seizures every day. He couldn't feed, and he needed help with his breathing. His EEG, his brainwave tracing, was markedly abnormal. 35 years ago, we wouldn't have been able to stop Levi's seizures, and he would have had really severe developmental impairments. Most children with his type of epilepsy would never sit or walk. I got into this field about 15 years ago because I was really frustrated by how little we could do for children like Levi. But between 35 years ago and when I started, there had actually already been some significant advances in our understanding of the reasons for these conditions through advances in genetic technologies. Now, standing next to me is Sebastian Lunke. Sebastian is an associate professor and he heads up the genomic division at the Victorian Clinical Genetics Service. Seb, you've been really heavily involved in this field for a number of years now. Can you walk us through what changed between 35 years ago and when you and I started in our fields?
Yes, Catherine. Let me start off by telling you a bit more about what I actually do. My lab is relying on a technology called DNA sequencing, which is essentially a very expensive way to look for typos in the blueprint that makes up the human body. Now, when MCI and VCGS started out about 35 years ago, that technology was still very much in its infancy. Back then, scientists like myself had to manually and painstakingly separate individual bases of DNA on gels, take photos and count. Try that for all 3.2 billion bases in the human genome. It's really like trying to visit every country on Earth while riding a horse. Back then, we didn't even have a full human genome sequenced. It is only with the first revolution in DNA sequencing technology, a technology called Sanger sequencing, that we were able to start looking at conditions, genetic conditions that affect children. But we didn't even have a human genome sequence available 35 years ago. Only the emergence of Sanger technology allowed us to obtain that crucial piece of data that we now use to compare the DNA of healthy children to sick children like Levi. This equipment was standard in every genetics laboratory when I started out 15 years ago, and even today is still used as standard of care to look at small pieces of DNA and find things that make children sick. Would that have helped Levi?
Unfortunately, no, Sanger sequencing 15 years ago wouldn't have been enough. And the reason it wouldn't have been enough is that we actually only knew of a handful of genes for epilepsy at that point. And as you said, with Sanger sequencing, you needed to know exactly where to look. And at that point, we didn't know of any genes for the particular type of epilepsy that Levi has had. So sadly, if he were born 15 years ago, his treatment would have been the same as 35 years ago. But that's not the case today, and one of the big reasons for that is really the sort of massive revolution, I guess, in our knowledge that's come about over the last 15 years. And so compared with 15 years ago, we now know of about 400 genes for genetic epilepsies. And this has been a really big game changer because it means we can now give families answers about why their child has had the problems and symptoms that they have had. What would it have meant for Levi though? So if he were born 15 years ago, he probably would have been enrolled in research genetic testing. And as we found more and more causes, we would have eventually found his, and he might have been 8 or 10 years old. And by that stage, it would have been too late to help him. It's very different now to what it was 15 years ago. And that has all happened because of the second revolution, I guess, in genomic technologies.
Yes, see, the problem with Sanger sequencing, Catherine, is that it can only look at very small pieces of human DNA at any one time. So while it was indeed used to sequence the first complete human genome, that effort required hundreds of these machines, took 10 years to complete, and cost over $1 billion. It is really like having a car. You can visit every country in the world, but it's really not practical. To help children like Levi, we needed the second revolution in DNA sequencing technology, the emergence of so-called next-generation sequencers. These machines are now able to sequence dozens of human genomes in a matter of days for only a few $1,000s each. It's like traveling in a jumbo jet. But even that is not fast enough. Critically ill children like Levi need answers in days, not months. So what we really need is a fighter jet. And that is exactly what we built. Our acute care genomics project, which is co-led by Professor Sonitsa Stark and myself, is now delivering genomic test results for children all across the country in under 3 days. Levi was one of over 350 children that participated in the project. What did receiving his answer in under 3 days mean for him, Catherine?
It was a game changer, a massive game changer. Through the project, Levi was 2 weeks old when he got his diagnosis. And this meant that we could offer his parents, Kasha and Steve, a lot more than just an answer. We don't have a cure for his condition, but we could use the genetic diagnosis to guide our seizure treatment. We started a new medication and his seizures stopped. And instead of having hundreds more seizures, which is what we would have expected, he's had only 3. This is Levi on his first birthday. Through the Acute Care Genomics project, we've had 7 children diagnosed with exactly the same genetic condition as Levi. Every single one has had a treatment change, and they're all doing better than we expected. We've seen the benefits of rapid genomic testing across many childhood conditions, not just epilepsy, but we've still got quite a way to go to understand its full potential and to have it fully available to all children with suspected genetic conditions. Recently, we've teamed up with 3 other world-leading paediatric institutes, Boston Children's Hospital, Great Ormond Street in London, and the Hospital for Sick Kids in Toronto, to form the International Precision Child Health Partnership, or IPCHIP. IPCHIP's goal is to enable experts in child health and genomics to improve precision diagnosis, implement personalised treatment decisions, and develop novel treatments. And the partnership's first project is going to be one in infantile epilepsies. And we're really excited to have the collective might of these institutes to drive us all forwards. Seb, what do you see as the next revolution in genomic technologies?
What we have done over the last 35 years, I think, is incredibly exciting. Technology has progressed so much that we can now help many children with suspected genetic disorders, from neurological disorders like Levi's to metabolic disorders, heart conditions, and many others. But I think we have also seen that the faster we get an answer, the better the outcome for the child. So just imagine a future in 35 years where we can look at a child's DNA before they even get sick. I think 35 years from now, we will sequence every child's genome at birth, or even while they're still in the womb. That data will be used in their healthcare throughout their life. All of a sudden, we could have diagnosed Levi in a matter of hours, not days. In other cases, we may be able to predict conditions that affect individuals in the future when they are adults. We may even be able to predict conditions that affect their future children. The technology is here now. We just need the support to start using it. But not only will we be able to predict conditions, we will also be able to tailor treatments to the individuals and give them exactly what they need.
That's right.
That is one of your key areas of interest, Catherine, isn't it?
Yeah, absolutely, that's the goal. So we're very much in the infancy of the era of precision treatment, as we call them, personalised treatments, and certainly what we could offer to Levi was a personalised treatment, and we've seen the wonderful impacts of that. But to some degree, even that is just a band-aid, because really it's a treatment directed to the symptoms and not to the underlying cause. There's a lot of work going on around the world to develop treatments that are directed to the underlying cause and that hopefully will be able to cure these conditions. And I hope that in 35 years' time, we'll be able to offer all children with conditions like Levi's and many other genetic conditions a cure for their conditions. And hopefully rapid diagnosis will mean that we're in a position to implement them as early as possible when they'll have the most benefit. We'll go now to Kasha and Steve, who are going to tell us in their own words what a rapid genetic diagnosis meant for Levi and their family.
Hi, my name's Steve and this is my wife Kasha, and this is our 22-month-old son Levi. Levi was diagnosed with a rare genetic condition of epilepsy at the age of 3 weeks old called KCNQ2.
We just wanted to thank the Murdoch Children's Research Institute and Dr. Catherine Howe for the early intervention that they provided.
Without their support and guidance and all their research, it could have been months or years before Levi got his diagnosis. So to be able to get that in the first 3 weeks of his life and then prescribe the right medication to help with his seizures was a big relief to us, and we're really thankful and privileged to be able to be with Dr. Catherine Howe and all the Institute's support along the way. Without their support and guidance, we don't know where we would be today.